Hello students, good morning. Last class we studied about one dreaded disease. What is that? Yes, it is AIDS. We studied about AIDS. In what way it spreads from one person to another and how does it replicate inside our body. What are the measures taken by WHO to prevent the spread of disease. All these concepts we studied under the heading AIDS. Today we are going to learn about one more dreaded disease, that is cancer. Cancer is one of the most dreaded disease of human beings and is a major cause of death all over the world and globe. More than 1 million Indians suffer from cancer and a large number of them die from it annually. The mechanism that underlie development of cancer are oncogenic transformation of cells. The development of cancer, we can call this as oncogenic transformation of cells. That means oncogenic means connected with cancer, and transformation, changing our cells. Our cells, in what way our cells are changed into cancer cells, and its treatment and control have been some of the most intense area of research in biology and medicine. So nowadays research is going on to find out how does a cell change into a cancer cell, and also what are the treatments and how can we control all these areas. Research is going on nowadays. Now in our body, first of all, we have to know the normal growth of our cells, body cells. In our body, the cell growth and development is regulated and controlled. So our body cells, they are well disciplined. Their growth and their differentiation is controlled and regulated. That is normally. And when normal growing cells, when the cells are growing, when these normal cells, they come in contact with other cells, when they grow in size and then they come in contact with the other cells, that time with other cells their uncontrolled growth is inhibited. That's why afterwards they won't increase in number or increase in size. Their growth is inhibited, that means stopped. Okay, that is the normal cell growth. But cancerous cells, this is known as contact inhibition. That means our cells, when they come in contact with the other cells and they stop growing, and that property is known as contact inhibition. This is the property of normal cells in our body. But these cancerous cells, they lost this contact inhibition property, and they continue to divide and give rise to a mass of cells known as tumors. Tumors means a tissue, a mass of cells. So when these cancer cells, they lose the property of inhibition, that is contact inhibition property, they continue to grow and then they produce a mass of cells because number increases and the size also increases and they produce a mass of cells, and that is known as tumors. Tumors are of two types, benign tumor and malignant tumor. Benign tumors, normally they remain confined to their original location and do not spread to other parts of the body, and also they cause little damage. So benign tumors, they remain in one place and they do not spread, and also they cause little damage to the body. On the other hand, these malignant tumors are a mass of proliferating cells called neoplastic cells or cancer cells. These cells are called, malignant tumor cells are called neoplastic cells or cancer cells. And then these cells, they grow rapidly. They grow very rapidly and they invading and damaging the surrounding normal tissues. They grow rapidly and they damage the surrounding cells. As these cells actively divide and grow, they actively divide and then they grow, they also starve the normal cells by competing for vital nutrients. They take away the nutrients, and so other normal cells, they starve of nutrients. They won't get nutrients. The cells that these neoplastic cells of malignant tumor, they mix with blood, and then they reach different distant sites through blood, and wherever they are, they start a new tumor there. So the cells that the malignant cells, they mix with the blood, and along with the blood flow, they go to different parts of the body, and wherever they get lodged or wherever they stay, there they start one tumor, and this property is known as metastasis. This feared property, dangerous property is known as metastasis. Okay, and next, shall we learn about the causes of cancer? How does this cancer cause in our body? Transformation of normal cells to cancerous cells is by different agents like physical agents, chemical agents, and biological agents. These agents are known as carcinogens. So the transformation of normal cells into cancerous cells is by many agents, physical agents, chemical agents, or biological agents. These agents that bring about cancer are known as carcinogens. So cancers are caused by carcinogens. Ionizing radiations like X-rays and gamma rays and non-ionizing radiations like UV, they cause DNA damage. They damage the DNA and leading to neoplastic transformation. Neoplastic transformation means what? Changing the normal cell into cancer cells. So ionizing radiations, X-ray and gamma rays, they also cause cancer. Non-ionizing radiations like UV rays, they also cause cancers. And chemical carcinogens present in tobacco smoke have been identified as a major cause of lung cancer. So the chemical carcinogens present in tobacco smoke, they cause lung cancer. And then oncogenic viruses. Some viruses called oncogenic viruses, they have genes known as virus oncogenes. They are viral oncogenes. They also cause cancers. And then several genes called cellular oncogenes. Cellular oncogenes or proto-oncogenes. Proto-oncogenes have been identified in normal cells. Our normal cells, they have cellular oncogenes and proto-oncogenes. And these cellular oncogenes and proto-oncogenes present in the normal cells, when they are activated under certain conditions, could lead to oncogenic transformation of the cells. That is transformation of normal cells to cancer cells. Okay, so what are the different agents that cause cancer? That is ionizing radiations like X-rays, gamma rays, and non-ionizing radiations like UV rays. And then chemical carcinogens also cause cancers. And then viruses, some viruses, that is oncogenic viruses, they also cause cancer, and they have viral oncogenes. And sometimes our normal cells, they have cellular oncogenes and proto-oncogenes. When these two are activated under certain conditions, they also lead to cancer. So these are the different causes of cancer. Now we will study about the detection and diagnosis of cancer. How can we detect the presence of cancer in our body, and diagnosis, that is also detection. Okay. Now early detection of cancers is essential. Early detection is essential as it allows the disease to be treated successfully in many cases. So if we detect the cancer earlier, we can even cure the patients out of this cancer. And detection is based on biopsy and histopathological studies of tissues, and then blood and marrow test for increased cell count. So we can detect the presence of cancer by biopsy. We will study each and every thing separately. Biopsy and histopathological studies of tissues, our tissue, studying our tissues, and also number of cells present in the bone marrow, whether more number of cells are present in the bone marrow or not, we will study. And also by this also we can detect the presence of cancer, and also techniques like X-ray, CT and MRI, CT scan and MRI scan and X-ray. These techniques also help us to detect the presence of cancer or diagnosing cancer. What is biopsy? A piece of suspected tissue is cut into thin section and it is observed under the microscope. So in in biopsy, they cut a piece of suspected tissue and into a thin section and then examined under microscope by a pathologist, and they find out whether the cells are normal or they are changed into oncogenic cells. Oncogenic is also related to cancer, and neoplastic is also words also are related to cancer. And then what is a CT scan? That is computed tomography. CT means computer tomography. Here in CT this technique uses X-rays, and then we get 3-dimensional image of internal organs. So we can find out whether any changes happens in the organs. And then MRI, full form of MRI is magnetic resonance imaging. Magnetic resonance imaging, this uses strong magnetic field and non-ionizing radiations to accurately detect pathological and physiological changes in the living cells. By MRI scan, we can find out any pathological and physiological changes, that whether there are any changes in the living cells. And in this way we can detect the presence of cancer cells inside the bodies. And one more way by which we can detect the presence of cancer, that is antibodies against cancer. Antibodies against cancer specific antigens are also used for detection of cancer, some certain cancers. So antibodies are used. Antibodies against certain specific antigens, they are also used to detect the presence of certain cancers. And then one more technique, that is molecular biology, techniques of molecular biology. They detect genes with inherited susceptibility to certain cancers. This biotechnology, it helps us to find out whether any cells will in future will change into cancerous cells. So such individuals can be advised to avoid exposure to carcinogens like tobacco smoke in case of lung cancer. So this molecular biology, techniques of molecular biology help us to find out whether a cell in future will develop into a cancerous cell. Then we can advise the individuals, so be away from the carcinogens like tobacco, don't smoke tobacco in case of, if they smoke, then they will get lung cancer. We can advise them. So these are all the different diagnostic method for finding out the presence of cancer. And then next we will study about the treatment of cancer. Treatment of cancer. The common approaches for treatment of cancer are surgery, radiation therapy, chemotherapy and immunotherapy. So these are all the treatments: surgery, radiation therapy, chemotherapy and immunotherapy. One by one we will study in detail. Surgery, what is surgery? They will remove the cancerous tissue, that's all. But radiation therapy, in radiotherapy, tumor cells are irradiated lethally taking proper care of the normal tissue surrounding the tumor mass. So they take care of the normal cells and only the cancerous cells are irradiated lethally and they are killed by these radiations. By chemotherapy, the treatment is given by using drugs. Drugs are used to kill cancer cells, but these drugs are specific for particular tumors. So treatment can be done by surgery and then radiation and then chemotherapy. And this, the side effect, if we take the drugs for cancers, they have some side effects like hair loss and anemia. Anemia and hair loss you will get. And then sometimes most cancers are treated by combination of all the above three. Combined all the surgery, radiotherapy and chemotherapy. And one more, the last treatment is immunotherapy. It's very interesting. Immunotherapy. Normally our immune system, it has the capacity to detect any other foreign cells in our body and protect our body against the pathogens. But these tumor cells, what they will do, they hide themselves from the vicinity of the immune system. The tumor cells, they avoid detection and destruction by immune system. If the immune system, if they find out the tumor cells, our immune system will destroy the tumor cells. So these tumor cells, they avoid detection, they hide themselves from the immune system. So what we do, the patients are given alpha interferons. If alpha interferons are given to the patients, these alpha interferons, they activate our immune system and help our immune system in detecting and destroying the tumor cells. These alpha interferons are also known as biological response modifiers. So when these alpha interferons are given to the patients, then our immune system becomes activate and it finds out our tumor cells inside the body and it destroy. In this way, the immunotherapy, it plays an important role in treating cancer cells. Okay, I hope all of you might have understood all the concepts under the topic cancer. Then next video I will teach you about drugs and alcohol abuse. Thank you, students.